Hi, students. Are you someone who opens up a practice lab test from Blackboard and then takes a look at some of the slides and stares at the models exactly where the structure has an arrow and a number, of course, such as here, here, and here? And then you go and take a look at the bottom notes, the PowerPoints, and try to memorize all the words that go with the numbers that look like the picture. Is that you? If it is, raise your hand. Okay? If it's you who's raising your hand or thinking about raising your hand, stop. Because I have another approach for you to try. I often hear students say, I don't have any time. So I hope that this approach can be more effective and efficient in your utilization of time with cadaver pictures, such as this one of the kidney, as well as with artist hand-drawn pictures, such as this one, and of course with models, such as those in your practice PowerPoint slides, which are also located in your lab for you to touch and manipulate and turn around and think about the structures in that manner, that tactile sense as well. Of course, the goal with any of my instructions is always to try it in order to improve your learning as well as to prepare for making better grades on tests. Okay, before we get to the microscopic level of a kidney, let's take a look at the gross structure of a kidney. You need to be able to identify or visualize what the kidney looks like in this case at the macroscopic or large or gross level before you can begin taking a look at smaller details of the kidney. So the kidney is a kidney bean shape. Of course, here we are looking at just one single kidney. But we know that there are two kidneys. They are located on each side of the body, laterally and deep behind the peritoneum. Hence the term retroperitoneal. Here's one more concept to consider before we move on to the renal blood flow. This is a decellularized kidney from Wake Forest Institute of Regenerative Medicine. And what a decellularized kidney is a kidney in which all of the cellular structures and DNA have been washed away, only leaving the local extracellular matrix, which includes polysaccharides and fibrous proteins. This image leads me to discuss the concept of 1,000,000 nephrons. Remember from your reading that there are 1,000,000 nephrons in each kidney. So you can think about this concept here as well. This is an interlobular artery, which is represented on this decellularized kidney as the endpoints of all of these arteries here. So if you can imagine the endpoints of all of these red tips that show interlobular arteries, such as the one in this hand-drawn illustration, then you can visualize the concept of 1,000,000 nephrons. This is a drawing of just one nephron that is connected to an interlobular artery. So here again is the decellularized kidney, and at the tip of each of these interlobular arteries, they're seen more clearly here against the white background, is a nephron. So this is the concept of 1,000,000 nephrons. However, we are going to study nephrons in isolation because 1,000,000 is just a little bit difficult to wrap my brain around. Students, stay tuned for part 2 of Learn How to Learn Anatomy and Physiology using lab models and pictures, and know that you can do it.
Sing it.
Yes, she can. Yes, she can. Oh, yes, she can.
Oh,
Yes, she can. Yes, she can. Oh, yes, she can.
They thought that I'd be weak.
But you made me strong.
They thought that I would crumble.
But they were so wrong.
You held my hand.